Good morning. In this video, we will be discussing about the biochemical aspects of porphyria. This is the pathway by which heme is synthesized in our body. Whenever there is a defect in this pathway, heme cannot be synthesized. Normally, heme acts as an inhibitor for the enzyme ALA synthase, which catalyzes the first step in heme synthesis. Succinyl-CoA will condense with glycine to form aminolevulinic acid. The enzyme for this reaction is ALA synthase. This enzyme will be inhibited by the pathway's final product heme. Okay, so thus it helps in regulating the enzyme. So normally the heme produced in this pathway will be inhibiting this step. But whenever there is a defect in this pathway and heme is not produced, what will happen? Heme will not be there, so the inhibition will be relieved. So what will happen means the production of the intermediates like uroporphyrin, coproporphyrin and protoporphyrin will increase. They will be accumulating in the blood and it leads to a condition called as porphyria. Porphyria is nothing but accumulation of porphyrin and other intermediates due to the defect in heme synthesis pathway. This is the definition for porphyria. This porphyria can be of two different types. One is congenital and the other one is acquired. In congenital porphyria, the disorder will be due to congenital deficiency of the enzymes, whereas acquired porphyria are caused due to all these causes. To start with, lead poisoning, then vitamin B6 deficiency, which is usually caused by ethanol poisoning and by the drug anti-tubercular drug isoniazid, iron deficiency, chronic renal failure, liver diseases and certain malignancies. All these can lead to acquired porphyria. Among these, the most important one is lead poisoning. So this lead poisoning usually inhibits ALA dehydratase and ferrochelatase enzyme involved in heme synthesis pathway. Normally, both these enzymes, they require metal ions for their action. ALA dehydratase, it requires zinc, whereas ferrochelatase, it uses iron for its action. However, during lead poisoning, the excess lead present in the blood, they will go and displace these metal ions, they will bind with the enzyme and they will inhibit the pathway and it leads to acquired porphyria. So these are some of the major causes for acquired porphyria. Next, coming to the congenital porphyria, this can be further subclassified into three types based on where the enzyme is deficient. Normally, heme synthesis, it takes place mainly in two sites. One is within the RBCs, second within the hepatocytes. In the RBC, it will be utilized for hemoglobin synthesis. Whereas within the liver, it will be utilized for cytochrome synthesis, which in turn will be utilized for detoxification and other metabolic process. So the heme synthesis, it takes place majorly in RBCs and in liver. If the enzyme is deficient within the RBCs, it leads to erythropoietic porphyria. If the same enzyme, if it is deficient within the liver, it is called as hepatic porphyria or non-erythroid form of porphyria. Sometimes the enzyme defects may be seen in both these tissues, that we call it as combined form of porphyria. Among these three different types, the most commonest form is hepatic or non-erythroid form of porphyria. That is the reason in some books it is mentioned that porphyria will it will not be associated with anemia. Why? Because the deficiency is commonly seen within the liver cells. So hemoglobin synthesis will not be affected. That is why porphyria will not be associated with anemia. But if it is erythropoietic porphyria, it will be definitely associated with anemia. Okay, so this is the classification based on the site of lesion. Okay, next, based on the exact enzyme deficiency, these porphyrias are further classified into 8 different types. So these are the 8 different types of porphyria, congenital porphyria with their corresponding enzyme deficiencies. Okay, so in this, the most commonest form of congenital porphyria is acute intermittent porphyria, which is caused by the enzyme deficiency of porphobilinogen deaminase. There are two other names for this enzyme. What are they means? One is hydroxymethylbilin synthase and the other one is UPG1 synthase. Okay, uroporphyrinogen 1 synthase. So all these are different names for the same enzyme. Okay, so when this enzyme is deficient, it leads to a condition called as acute intermittent porphyria. Okay, so this enzyme name, you should memorize it, no other go. But for few other enzymes, I can give you small clues. So first, to start with ALA synthase. Here the enzyme name, the second name of the enzyme, it starts with S. So similarly, the disorder also, it starts with S. Okay, so the deficiency of ALA synthase, it leads to a condition called as sideroblastic anemia. Okay, since it is an erythropoietic variety, it is presenting with anemia. That's why this condition is called as sideroblastic anemia. Next, coming to UPG co-synthase. Here, the second word of the enzyme, it starts with C. Similarly, the disorder also, it starts with C, which is nothing but congenital erythropoietic porphyria. Congenital erythropoietic porphyria is caused by the deficiency of the enzyme UPG3 co-synthase. Next, CPG oxidase enzyme. What is the function of this CPG oxidase? Means the name itself says it oxidizes CPG, coproporphyrinogen will be oxidized. Suppose if this enzyme is defective, this CPG cannot be oxidized, so CPG will be accumulating. That is why this condition is called as coproporphyria. Coproporphyrinogen will be accumulating. That's why it is called as coproporphyria or hereditary coproporphyria. Next, coming to the last enzyme, furochelatase. So what is the action of this enzyme? Means it converts protoporphyrin to heme. That is its function. The last step in here, heme synthesis, is catalyzed by furochelatase. Okay, when this enzyme is deficient, heme cannot be formed. On the other hand, protoporphyrin will be accumulating in the blood. That is why this condition is called as hereditary protoporphyria. Okay, so then again one more enzyme, the ALA dehydratase. That is very simple, easily you can remember, because its deficiency leads to ALA dehydratase deficient porphyria. Okay, so for the remaining three enzymes, you have to memorize the name, no other go. Okay, so PBG deaminase, it leads to acute intermittent porphyria, which is the most commonest form of porphyria. Next, UPG decarboxylase, it leads to Porphyria cutanea tarda, which is the second most common form of porphyria. And finally, PPG oxidase enzyme deficiency, it leads to Porphyria variegata. Okay, so these are the different types of porphyria which you can see. Next, we will see how to diagnose all these different types of porphyria. So during porphyria, the porphyrins and other intermediates will be accumulating within the RBCs. Suppose if it is a erythroid variety, it will be accumulating in the RBCs. Suppose if it is a hepatic variety, it will be accumulating in the plasma. Okay, so this accumulated products, intermediates, they can be excreted from our body either through urine or through feces. The major porphyrins that will be accumulating there are three major porphyrins. One is uroporphyrin, second coproporphyrin, and the third one is protoporphyrin. These are the three major porphyrins that can be accumulated during porphyria, during condition called as porphyria. Okay, so in this uroporphyrin, they will be having acetic acid and propionic acid side chains in the structure. Okay, so both these acetic acid and propionic acid are water soluble, so they can easily dissolve in urine and it can be excreted through the urine. Next, coming to CPG, coproporphyrinogen. Here, the acetic acid group will be replaced by a methyl group. Okay, so that means CPG will be having methyl group and a propionic acid group as a side chain. So this propionic acid is a water soluble, whereas this methyl is a water insoluble compound. That is the reason this CPG can be excreted both in urine and also in feces. Next, coming to the third one, protoporphyrinogen. Here it will be having more methyl side chain and mostly vinyl side chain. Okay, so both this methyl group and vinyl group, they both are water insoluble. That means it cannot be excreted through urine and it can be found only in the feces. Okay, so this is how various uroporphyrins, the intermediates, will be excreted or removed from our body. Okay, so from all these samples, these porphyrin intermediates, they can be detected by various qualitative methods and quantitative methods. First, we will see about the qualitative methods. Qualitative methods means it will just help to detect the porphyrin, presence of porphyrin. It will tell you whether the porphyrin intermediates are present in the sample or not. Quantitative means they will be helping in quantifying it, by measuring the amount of porphyrins excreted or present in all these samples. So first we will see about the qualitative test. So the most commonly used qualitative test for diagnosing porphyria is UV fluorescence test. When you pass UV light to the sample, the porphyrins within the sample, they will be absorbing the UV light and they will get excited. Once it gets excited, they will be releasing fluorescence that we can easily measure and we can detect the presence of porphyrins. Okay, so this is the most specific method for detecting porphyria, for detecting porphyrins and for diagnosing porphyria. Next, apart from observing UV light, the porphyrin rings can also observe for light of 400 nanometer. Okay, so this can also be identified using another technique called as spectroscopic technique. So when you visualize the sample through this spectroscopy, what will happen means you will be getting a characteristic dark band at 400 nanometer called as Sorad band, which is named after the scientist who discovered it. Okay, so the porphyrins present in the sample, they will be observing the 400 nanometer. Okay, so in the spectrum at 400 nanometer, since that light has been observed, you will be getting a dark band there, which can be used for diagnosing porphyria. Okay, so apart from these spectral techniques, we also have few chemical methods to detect the presence of porphyrin intermediates in the urine. Okay, so the most commonly used method is Ehrlich's test. Okay, the Ehrlich's reagent, dimethyl ammonium benzaldehyde. Okay, so dimethylamine benzaldehyde, that will be used for detecting the presence of porphyrins and other intermediates. Okay, so these are the various qualitative tests by which we can diagnose porphyria. Next, coming to quantitative method, we can use spectrophotometric estimations. Okay, we know that it can observe UV fluorescence and also the light of 400 nanometer. By using any one of these things, we can measure by the intensity of the light how much it is absorbing. Based on that, we can easily calculate the concentration of the porphyrins present in the sample. Okay, so all these things can tell you that the patient is suffering from porphyria. But if you want to identify what type it is, you have to go for the enzyme studies. Okay, so you have to measure the enzyme level either in RBCs or in the liver. Okay, if it is an erythroid form, go for the enzyme levels in RBC. If it is an hepatic form, go for the enzyme levels within the liver cells. Okay, so or ultimately you can also go for DNA study to identify the mutations which leads to all these congenital porphyrias. Okay, so that's it about the diagnosis of porphyria. Next, we will see about some of the few important individual porphyrias. Okay, so these are the various porphyrias and their enzyme deficiencies. So all these congenital porphyria, they again can be classified into two types based on the clinical features. Okay, so this is the demarcation. So any enzyme deficiency above this line. Okay, so any enzyme deficiency above this line, that means deficiency of PBG deaminase, ALA dehydratase and ALA synthase. When these three enzymes or any of these three enzymes are deficient, there will be accumulation of ALA and PBG. So this excess ALA and PBG, they result in neurological manifestations. Whereas if the enzyme deficient is below this line, that means any of these enzymes are deficient, it will be leading to the accumulation of porphyrins, UPG, CPG and PPG. The porphyrin, the characteristic porphyrins will be accumulating, which usually presents as photosensitivity reactions. Okay, so why there are neurological symptoms are caused and why photosensitivity reactions are caused in some porphyria, I will explain you by taking example from each one of this class. Okay, so from here, let me take this acute intermittent porphyria, which is actually the most commonest form of porphyria. Okay, so about each one of these individual porphyria, you should normally know 4 facts. What are those 4 facts? Means first thing is site of enzyme deficiency. That means whether it is erythroid form or it is hepatic form, that you should know first. Second, what is the exact enzyme deficiency? That is the second thing you should know. Okay, what is the exact enzyme deficiency? Third, you should know the major clinical features you will be seeing in the condition. Then fourth is how to diagnose this condition. Okay, so these are the 4 facts you should know about each one of the porphyria, various porphyria. Okay, so now acute intermittent porphyria also I will be discussing under the same headings. To start with, site of deficiency. So this site of deficiency you can easily remember. I will give you a small clue to remember this. The first disorder, okay, the first enzyme disorder, that means the deficiency of ALA synthase, which leads to sideroblastic anemia. Okay, so the first disorder and the last disorder. Okay, the last disorder is deficiency of ferrochelatase leading to hereditary protoporphyria. Okay, so first disorder, last disorder and the middle disorder, congenital erythropoietic porphyria, which is caused by UPG3 co-synthase. Okay, so these 3 disorders, they are erythropoietic varieties, whereas all the other disorders are hepatic varieties. Okay, very simple to remember. Okay, so first one, last one and the middle one. Middle one you can easily remember because the name itself says it is congenital erythropoietic porphyria. That means it is an erythropoietic form. So apart from that, you should also remember the first and the last thing. Okay, so these 3 disorders, they are erythropoietic form, whereas all the others are hepatic forms. Okay, so first to start with acute intermittent porphyria, it is hepatic form. Okay, so the enzyme is deficient in the liver. Okay, so what enzyme is deficient means here it is PBG deaminase, that is the enzyme deficient here. Okay, so the PBG deaminase, porphobilinogen deaminase enzyme will be deficient. Okay, so this enzyme, it has also two other names. What are the other names means? Hydroxymethylbilin synthase, or otherwise called as UPG1 synthase. Okay, so these are the various names for the same enzyme. So when this enzyme is defective, it leads to acute intermittent porphyria. Coming to the clinical features, so whenever this enzyme is deficient, what will be accumulating? You can see the pathway here. Okay, so when the pathway PBG deaminase is not produced, what will happen means the precursors PBG and ALA, aminolevulinic acid, and porphobilinogen will be accumulating in the blood. So this ALA and PBG, they mainly lead to neurological manifestations. So how it leads to neurological manifestations means ALA, the aminolevulinic acid, they usually inhibit the ATPase pumps present in the nervous tissues or in brain, resulting in conduction paralysis. That is the reason these patients, they will mainly present with symptoms, neurological pains, okay, mainly abdominal pain, and also like psychiatric symptoms. Okay, so these will be these are the major clinical manifestations you will be getting in not only acute intermittent porphyria, all the three porphyria above the demarcation line. That means above acute intermittent porphyria, all the three disorders, you will be getting mainly these neurological symptoms. Okay, next coming to the diagnosis part, here porphyrins will not be produced. Okay, so UPG, CPG, PPG will not be produced. Okay, because PPG deaminase is deficient. So if that is not present in urine and feces, nothing will be elevated. So nothing will be excreted and nothing will be elevated. So you have to go for the plasma level. Okay, you have to measure the plasma levels of ALA and PBG. So both these things will be elevated in acute intermittent porphyria. Okay, so here actually I forgot to mention one important thing. So mostly in these patients, all these symptoms, neurological symptoms, they will be aggravated, especially after they are exposed to estrogen, barbiturates, or they are exposed to starvation. Okay, so why all these things are aggravating or precipitating the symptoms means, so these things, they actually have the tendency to induce the enzyme ALA synthase within the liver. Okay, ALA synthase. So if that enzyme is increased, what will happen? ALA production will increase. So when this increases, then all neurological manifestations will be increased or exaggerated. Okay, so that's it about acute intermittent porphyria. Okay, so in acute intermittent porphyria and the disorders above that, you will be mainly having neurological symptoms. Okay, so why you are having neurological symptoms? Because of the accumulation of ALA and PBG. Okay, next coming to the second type, which will be presenting with photosensitivity reactions. Okay, so in that, let me take congenital erythropoietic porphyria and I will explain you the how it manifests. Okay, so this congenital erythropoietic porphyria is quite unique from the remaining types of porphyria. How it is unique means, all the other congenital porphyrias, they are autosomal dominant inheritance. They will follow autosomal dominant inheritance. Whereas this is the only porphyria which follows autosomal recessive pattern. Okay, so that's why I said it is an unique form. Okay, so regarding this congenital erythropoietic porphyria also, I will be discussing under the same four headings. Okay, so to start with, site of lesion. So here the name itself says it is congenital erythropoietic porphyria. Okay, so here it is an erythroid form of porphyria. Okay, so not only this, the first disorder and the last disorder. Okay, so they are also belongs to this erythroid type. Next, second, the exact enzyme deficiency. So this is caused by the enzyme defect called as UPG3 co-synthase. UPG3 co-synthase. What is the action of this UPG3 co-synthase means? It prevents the formation of UPG1 and facilitates the formation of UPG3. That is the function of UPG3 co-synthase. It prevents UPG1 synthesis formation and facilitates the formation of UPG3. Suppose if this enzyme is defective, what will happen? UPG3 will not be produced. Instead, UPG1 will be produced in excess. Okay, so you will be getting UPG1. From that UPG1, we can also synthesize CPG1. Okay, so uroporphyrins and coproporphyrins will be accumulating in the blood. So when these these porphyrins accumulates in the blood, it leads to, it mainly presents with photosensitivity reactions. Okay, so why it is having this why these patients are having this photosensitivity reactions means when uroporphyrin or coproporphyrin or protoporphyrin, when any of these porphyrins, when they accumulate, they will absorb light of 400 nanometer. They get excited, and this excited molecule, they will act on the oxygen and convert it into free radicals, and these free radicals will cause injury to the lysosomes, and these lysosomes will be releasing the degradative enzymes which can degrade the entire cell. So it leads to cell damage. Okay, so this type of cell damage, it will be most commonly seen in the skin layer, which is exposed directly to the light. Okay, so when light falls on our skin, on the skin of the patients with this type of porphyria, the uroporphyrins and coproporphyrins which are settled in the skin, they will be damaged. Okay, so they will be excited and they will be damaging the skin. That is why these people, they present with photosensitivity reactions. Okay, so the photosensitivity reactions will be, the skin damage will be very pathetic. Okay, you can see the skin will be damaged like this. Okay, so this is a very important manifestation you will be seeing in congenital erythropoietic porphyria. Not only in congenital erythropoietic porphyria, beyond this, okay, in all the other downgraded thing or enzyme porphyrias, the enzyme deficiencies will lead to accumulation of uroporphyrins or coproporphyrins or protoporphyrins. Okay, so whenever these porphyrins are accumulating, it presents with photosensitivity reactions. Okay, next, coming to the last diagnosis part here, when UPG3 is not there, what is happening? UPG1 is formed, and from that CPG1 can also be formed. Okay, so UPG1 will be usually excreted in urine, whereas CPG1 will be excreted in urine and feces. Okay, so we can easily demonstrate these two intermediates in urine and feces, and we can easily make the diagnosis. Okay, so that's it about congenital erythropoietic porphyria. Okay, so just for this example, I have taken two major types and I have explained you how the reaction, how the symptoms are happening. Even for the other things, you try to make a tabular column under these four headings. Okay, very simple. Site of deficiency, I have already told you. Okay, so first disorder, last disorder, and the middle one, congenital erythropoietic porphyria. All these three are erythroid forms, whereas all the remaining all are hepatic forms. Okay, so deficient enzyme, I have already given some clues, so based on that you can write it. Clinical features, PBG deaminase enzyme, and above it leads to neurological manifestation. It leads to accumulation of ALA and PBG, so they will mainly present with neurological manifestation. Okay, whereas after UPG3 co-synthase, from here and below, they will be mainly presenting with photosensitivity reactions due to the accumulation of uroporphyrins, coproporphyrins, and protoporphyrins. Okay, diagnosis part also we have already discussed. So try to read about all the other types of porphyria also under these four headings, but at least these two things you should be thorough with. Okay, so that's it about porphyria. Thank you.